soil awareness campaign that Globe Thailand have done. Before I start my talk, let me ask you to do something. Can you imagine that you are on space? Are you on space now? Okay, I think everyone now is on space. And if we are looking at the Earth from space, what do you see? Can you type? Imagine that you are on space and then what do you see?
No, nothing.
Nothing. Okay. You see?
Yeah.
Okay, I forget to say to you something that you look at the Earth from space. So we see land, water, cloud, atmosphere, snow, and plant, isn't it? Do you see anything else? Okay, let's zoom in. Come with me. Without plant cover, most of what we see is soil. Are you agree with me? Okay, soil is the keyword that I'm going to talk to you today, as Dr. Desh mentioned earlier. If you see in the online news, Facebook or on the Instagram that coming up 5th December is the World Soil Day. In 2002, the International Union of Soil Science made this day be a World Soil Day in order to celebrate the importance of soil as a critical component of the nature system and as a vital contributor to human well-being. Why 5th December? Do you remember what Dr. Det said before? The date of 5th December was chosen because it corresponds with the official birthday of King Bhumibol Adulyadej, King Rama V. He is King of Thailand, who was one of the main proponents of this initiative because he have done the benefit project about soil for Thai people. This is our king, the late king, King Rama V. You can see in the picture. This one. And the theme of World Soil Day in 2020 is Keep Soil Alive, Protect Soil Biodiversity. And the aim of the raising the awareness of the importance of the maintaining healthy ecosystem and human well-being by addressing the growing challenge in the soil management, fighting soil biodiversity loss and increasing soil awareness. Why do we need soil biodiversity? I need someone to help me. Okay, somebody answer. I'll try to read it. Why do we need soil biodiversity? Okay, I will share with you. The answer will be in this video. Okay, I will show you.
That distant and small spot in the cosmos is the habitat of approximately more than 2 million species, and the product of billions of years of evolution. Most of this biodiversity is in the soil beneath our feet, but only 1% of soil species have been identified so far. This soil biota constitutes the greatest concentration of biomass of any part of the planet and is composed of an immeasurable amount of mega, macro, meso, and microorganisms. Each of these wonderful creatures plays a key role in the food chain that maintains the fragile balance of life in natural and agroecosystems. If these complex interactions become interrupted, it can cause irreversible impacts for life on Earth, including humans. So, why do we need soil biodiversity? Soils hold many unexpected secrets and perform unseen functions. The discovery of antibiotics has had a major impact on increasing human life expectancy. Soils hold many other potential medicinal functions and possible cures. The natural association between plant roots and microscopic fungi promotes better plant nutrition and growth, tolerance to soil pathogens, and adverse climatic conditions. Through their natural function and metabolism, soil microorganisms are capable of breaking down and denaturing certain toxic compounds and contaminants resulting from many human activities. Part of CO2 emissions into the atmosphere derived from industry and agriculture can be absorbed by plants and stored in soils thanks to microbial decomposition that may allow soil carbon retention for long periods of time. This invaluable service provided by soil organisms is key in climate change mitigation. On the other hand, deforestation, monocultures, and the overuse and misuse of agrochemical inputs degrade and reduce soil's health, diminish resistance to pests and pathogens, and cause biodiversity loss that jeopardize the delicate balance that took billions of years to evolve and specialize. We could not only lose a long natural history of speciation, specialization, evolution, and adaptation, but also soil's capacity to perform essential ecosystem functions. How can we restore, manage, and conserve something that we do not see directly, and of which we do not have full knowledge? It is our collective responsibility to raise awareness on the importance of soil biodiversity, promote technological innovation to preserve and enhance soil biodiversity, including ecosystem restoration, recognize soil biodiversity as a key provider of ecosystem services, and as one of the main nature-based solutions to face all the current global challenges. Invest in gathering better knowledge about the status of soil biodiversity and functions, including by region and land cover type. And develop policies based on scientific evidence to mainstream sustainable soil management and conservation of rich biodiversity soils across landscapes. Everything that we eat and drink passes through the soil biota over and over again. Therefore, we need healthy soils for healthy food, healthy environments, healthy people, and a healthy life. It starts with you. Give a voice to the sound of a living soil. Keep soil alive. Protect soil biodiversity.
Okay. Keep soil alive, protect soil biodiversity. This video presentation is I took from Food and Agriculture Organization of the United Nations. And as you see in the video, to study soil, scientists need to be close up and in like personal. So when you measurement the physical and chemical of soil, and then our globe student, globe teacher, that you can share the data to globe observer that no one else can do, because you know the soil in your place or your area better, so you can help soil biodiversity and help scientists to see the soil. With the framework of the World Soil Day 2020, Globe Thailand is launching a soil awareness campaign on the soil biodiversity with the same theme is keep soil alive, protect soil biodiversity. And the aim of this campaign is to promote opportunity for the participants to promote scientific knowledge, raising the awareness of the threats posed by soil biodiversity loss, and stimulate educational activity engagement of young people and school in soil science. And the campaign is aimed at anyone who is interested in soil and soil biodiversity. This can include teachers, students, and public people. Participants can take part by they submitting the infographic or postcard that they address method to conserve soil biodiversity. Moreover, in each infographic that they wanna submit with us, they should contain picture of living creature in the soil. As you can see in the PR poster, the campaign has been in November 15. So I have received some infographic that I'm going to show you guys today.
The last question in timeout.
As you can see, because the infographic is from Thai
Those
people,
who have completed, they can write the next question.
The poster will be written in the Thai language, but I will give you some brief about each poster. This picture, the student want to present the benefit of
write the question and wait.
Soil and addressing the method to reserve the soil biodiversity by like application of organic matter to the soil, such as clog, stubble, support greater proportion of surface feeding creatures in earthworms.
Okay.
Next.
Who is that?
To picture the student
you can
want
see the
to show that earthworms can help create humus.
Okay.
Earthworms also help create good soil structure. Their burrows open up the soil and create aeration and drainage channels in the soil. I would like to add something. In addition, when the earthworms die, their protein-rich bodies return nitrogen fertilizer to the soil. For the last two sample, this picture say about if you live in a place without earthworms, the millipede can feel their need. Not only are they responsible for decomposing as much as like 10% of leaf litter in the garden, they benefit other soil microorganisms that work intimately together to turn their waste into nutrient-rich soil. For the criteria, most of participants will get the rewards if their infographic follow the requirements, and the requirement is not too difficult, because we want the student and people to join this campaign. Like the requirements are like submitting in the PDF form, A4 size, something like this, with the addressing method to conserve the soil biodiversity. Before I finish my presentation, there are some fun facts about soil I would like to share with you. You know, in just 3 inches of soil, there are 13 contributions. Condition mean 1,000 millions living organisms within 100 million tons. Amazing. 1 hectare of soil contains the weight equivalent of 2 counts of bacteria. That's a lot. And there are more organisms in 1 gram of healthy soils than there are people on Earth. And earthworm can digest its own weight in soil every 24 hours. 50% of the planet's soil pass through the gut of the earthworm each year. And soil organisms process 25,000 kilograms of the organic matter in the surface area, equivalent to soccer field, which is the weight of 25 cars. Okay, so I'd like to wrap up about the key message for my presentation today is soil is a living resource, home to more than 25% of our planet biodiversity. Use up 90%, up to 90% of the living organisms live and spend part of their life cycle in the soil. And it is estimated that only 1% soil microorganism species are currently known, compared to 80% of plant species. Moreover, soil organisms can break down certain contaminants, and soil organisms help soil store carbon. And have you know that New York's Central Park hosts more than 120,000 types of bacteria and over 40,000 species of fungi, protozoa, and others. Thank you for your attention.
Thank you, Nishi, for wonderful presentation. And we should really give thanks to her. This has been a wonderful presentation. The video was great. And this video will be uploaded on the website along with the presentation. And all our webinars are available on the website. And I have the link, or I can send, I will.
Research as a research project in the globe. As we know, next case aega. Hello. Archit. Archit. Idhar aao. How to share this? Next slide कैसे आएगा? अरे next slide कैसे आएगा यार? हेलो, ये उधर चला गया यार बार-बार. अरे बेटा तो इधर तो आओ आप लोग. अर्चित next slide नहीं आ रहा. Next slide is not coming. नहीं हो रहा क्या? हाँ. Okay.
ऐसे वापस पीछे जाने का बस.
अब तो हो रहा ना?
हाँ, अब हो रहा है.
Okay. As we know that in globe program, all the 4 spheres of the earth, that is hydrosphere, biosphere, atmosphere, and lithosphere, we study all the different spheres. So today we will be talking more about the lithospheres. We all know that these soil, which is very important, is a solid material, and there is a process of weathering and biological activity which makes the soil. The process of making the soil is called pedogenesis, and the scientific study of the soil is called pedology. It has come from the word pedon, that means soil or the earth. Now, in pedology, we study factors and processes of the soil formation, soil characteristics, and distribution of the soil types. In today's program, we will be talking only on the soil characteristics, both their physical characteristics as well as the biological and the chemical characteristics. Nisi has already told that 5th December all over the world is celebrated as a soil day, and for this year the theme is to protect the soil biodiversity. She has given very good examples of the bacteria and different kind of the organisms which are present in the soil. I'm just as a passing remark, I can say that even in 1 gram of the soil, there can be 10 to the 10 power 10 bacterial cells, and the diversity can be from 4.10 to 5.10 to the power 4 species. These may include the soil microorganisms, which may include bacteria, the actinoacetes, the fungi, algae, protozoans, etc., etc. And all these organisms remain near the root, and the root zone and the place near the root is called rhizosphere. So there are soil microorganisms also. These may include, as she has already told, about the earth forms. Then there can be many centipedes, millipedes, insects, and the spiders, and many more. Now, why students in globe should study the soil? In general, if we see, then we can see that in all the school syllabus, in classes of 8th, 9th, 10th, the soil is being studied. But in general, they study only theory. And in globe, we have a hands-on activity where the students learn by doing something. So soil is a foundation of the basic ecosystem. It filters our water, provide essential nutrients to our forest, grasslands, crops, and other, fruit plants, help regulate Earth's temperature, as well as they have different on the important for the soil greenhouse gases. Now, Earth's biodiversity, 25% of the total diversity of the Earth, biodiversity of the Earth, lives in the soil. And thus, they help in climate change and filter our water system. A healthy soil produces healthy crops. That means they just supply essential nutrients, essential water, oxygen, root support system, and the food which we grow, on which the whole earth is actually depending. Soil is a very important natural resource. It is a great integrator. It is, of course, helpful for crop production, producer and absorber of the gases, oxygen, carbon dioxide, and different greenhouse gases. They are medium for the plant growth. They are home for many organisms, including the micro and macro, and maybe that many burrow animals, they live inside the soil. One can study the geological past, that is climate, biological wealth, and human history. If we see the soil, it is a very big waste decomposer. It is a source for the medicine, art, culture, construction, and of course, a storehouse for the water. Now, in general, when we study the soil, we generally take a profile. that is a cross section, vertical cross section of the soil, where we can understand the different layers of the soils, and these layers are called horizon. The uppermost layer is called O horizon, where there is a more organic content present, and as we go below, there may be the layer of the topsoil, then at the end there may be the pellet material present. So one has to study soil profile, and preferably if we want to study the soil, its physical and chemical and biological properties, we have to take samples from different horizons so that we can understand the type of the soil, its physical, chemical and biological characters at different strata or different layers or different horizons. Now, there is a process of soil formation and we call it as pedogenesis. Pedogenesis means soil formation. The main is the parent material. The climate and organism act upon the parent material, taking its own time, and then convert, or there is a milling action, and then there is a pedogenesis, that is soil formation process, when the organic material goes inside the milled materials of the parent area. So soil in climate, the temperature, the rainfall, the withering and the humus, they play important role. The parent rock determines the color, the texture, the chemical properties, the mineral components and permeability of the soil. Relief actually depends upon the altitude, slope, and they determine where the soil will be accumulated naturally. If there is a plain area, more soil will be accumulated as compared to a hilly section. The flora and the microorganisms, they affect the humus formation process. And of course, as the time goes on, the thickness of the soil increases in a particular area. So these are the different parent rocks, the time, the biota, the topography, and of course the climatic actions. All these work together to form the soil. Now, the parent material, it actually is the initial stage of the soil system. And from the parent material, one can understand the soil color, the soil texture, the soil structure, its mineral composition, drainage, permeability, and all the other physical characters of the soil. If we see an ideal soil, which may not be available everywhere, but if we see an ideal soil, roughly 45% of the area of the soil is minerals, 25% water, 25% air, and maybe roughly 5% is the organic matter or the humus and the other organic material. Now, which site one should take for study the soil, especially the students of a school or a globe school. So we can take any site located anywhere which is safely exposed for the soil profile. It is very useful that it should be nearer to your soil moisture, temperature study site, or a land cover study site. So here the land cover study site has been shown, and there are many flags present here. One can see there are many flags present here, and these flags show that we have taken a star-shaped structure to soils for sampling the soil and its moisture. Now, how often one should conduct the soil protocols? Generally, soil do not change very quickly as the atmosphere and the hydrosphere characters used to change. So it depends upon the properties which you want to take. In general, the soil moisture content, local composition of air change over the periods of a few minutes or a few hours. Other properties may take very long time to change, like soil pH, soil color, soil structure, its bulk density, organic matter, microorganisms. So they may take a longer time. So in general, we should study the soil temperature and soil water content or the moisture more frequently, more periodically, maybe every month, maybe every week, maybe every season. But other characters one can study once a year or so. In soil protocol of the globe, we do study the soil structure, the soil color, the soil consistency, the soil texture, the soil temperature, the soil pH, the soil moisture, its bulk density, carbonates, of course, nitrates, etc. are also studied. So if we want to study the soil structure, we have to take a small amount of the soil in our hand. It is a hands-on activity. So you have to make your hands soiled. And if we study the different horizon of a soil, then the aggregate of the particles one should study from different horizons, upper horizon, middle horizon, the lowest horizon. Actually, the soil structure will say us or tell us that where there are air spaces and where there are the larger sized particles. A small pad of the soil is actually taken in hand. I will just take the first, just say, we take the small pad of a soil or a small clump of a soil, and we have to study it thoroughly. Now, for this or for digging, we need some kind of trowel or some kind of device, and we can identify the structure at different horizons. And these are the different kind of the structures. There are 7 kind of structure which has been in the globe protocol. There can be a granular soil. Now, these grains can be single grained, or there can be a soil which is prismatic, columnary, platy, blocky, massive. These are the different kind of the, so one has to see, the student have to take it into hand and then study it, and where it is a blocky or it is a prismatic or it is a granular or it is a single grain, one has to find out its soil structure type. Then second is soil color. Now the soil color is produced due to the minerals which are present in the soil and the organic matter which is present in the soil. So if there is a dark brown or black colored soil, it indicates that that soil has high organic matter content. In general, the wet soils will appear more darker as compared to the dry soil. And that's why for studying the soil and its color, we have to make the soil wet. Black or dark brown colored soil indicate the presence of decaying organic matter and shows that it is more fertile as compared to the brown or the yellow colored soil. So how to study the soil color? The soil color is always studied in the field. It cannot be studied in a room or in a classroom or in a laboratory. Moreover, the day on which you are studying, it should be a sunny day. And you take a small pad of the soil, spray some water on it with the mist bottle. Then you have to divide the pad into 2 parts and take the inner part of the, look on the inner part of the pad, and you stand against the sun so that your back is towards the sun and your hand is towards the sunlight. And then you have to compare the color from a standardized soil color book. Soil color book is approved by the globe protocols, and we have to take into account only that. So that was about the soil color. And there is a soil consistence. We have to find the consistence of the soil. Consistence means by how easily soil aggregates can break apart. So the soil can be loose, soil can be friable, soil can be firm, or soil can be extremely firm. So we have to take the soil in our hand and put it between thumb and the finger and just try to squeeze it or make a pressure on it, and then we can find out that how much it is, what is the consistency of a soil. Similarly, soil texture is mainly depending upon the different size of the soil particles which are present. We namely say them as sand, silt, and clay. Sand particles are large particles, silt particles are medium-sized particles, and the clay particles are the small-sized particles. So, soil texture is mainly depending upon the amount of all these three, that is sand, silt, and clay, while the consistency is depending upon the soil particle size only. So there is a classification system. Sand is 2 to 0.05 mm of size. Silt is 0.5 to 0.002 mm of the size, and the clay is less than 0.002 mm size. So this is how we define the sand, silt, and clay. So in nature, it is not essential that all the particles are sand, all are clay, or all are silt. So depending upon the percentage of the clay, silt, and sand, one can define the soil of different type. This is a soil triangle. If there is more sand, it is a sandy soil. If there is more silt, it is a silty soil. But it can be a sandy loam, it can be a clay loam, it can be only a loamy soil, it can be a silty clay loam, or any different kind of the classification one can understand or say. In general, loamy soil is considered as a good soil as it supports the different crops like climbers, bamboos, perennials, shrubs, tubers, etc. In general, if we want to sow the onion, carrots, or the root crops, there should be a more percentage of the sandy soil. Of course, if there is a more percentage of the sandy soil, it is very high, then maybe it lacks the water holding capacity and nutrient holding capacity. So if your soil is mucky clay, you can improve its texture and structure by adding some sand and some compost. The sand will quickly improve the texture by separating some of the smaller mineral particles and allowing opening of the air and water circulation. This is how we can study the soil. We have to take some sample from this is a surface sample only. We have not studied here the whole different strata or the different, depth of the soil. Here we are taking only from the surface. So we have to take the surface soil sample, and to know the texture, what one has to do, we have to moisten the soil. Once we moisten it thoroughly, you have to just rub it in your palm or in your this mist, and then you make a ball of the soil. In case this ball is not made, no ribbon is formed, no ball is formed, that means it is a coarse textured soil. If no ribbon is formed but the ball is formed, that means it is a moderately coarse textured soil. If there is no any small pieces are formed, then it is medium textured. So we have to form the ribbon, taking the ball in between your finger and the thumb, you have to form a ribbon. So this is called a ribbon method, in which one can very easily know the fine textured, moderately fine textured, medium textured. Now, another very important thing is soil. Soil temperature affects the seed germination and plant growth, because the roots grow inside the soil, and that's why different organisms also survive best and they function best at the optimum soil temperature. It also influences the soil moisture, aeration, ability of the nutrients availability for the plants. At a constant moisture content, a decrease in temperature results in a decrease in water and nutrient uptake. At a very low temperature, the transport from the root to the shoot is actually reduced. So, for measurement of the soil temperature, in globe, we use a dial type thermometer. This is the dial type thermometer. Now, you can measure the soil temperature by this thermometer at different depths. In general, in globe, we calculate or we observe the temperature at the depth of 5 centimeter and at a depth of 10 centimeters. Another is soil moisture. Now the soil moisture is very important for nutrient uptake, use of the water by the plants, atmospheric, it also affects the atmospheric humidity, and of course the flooding situation. And it actually, whenever there is a rain or there is a melting of the snow, there can be more soil moisture. But at the same time, the plantation at any place will support and will control the flooding of the water. So, why we want to know the soil moisture and why want to measure the soil moisture? It relates to precipitation, it relates to surface soil and air temperature. It also, depending upon the diurnally, it changes. In the morning, in the day, in the evening, the moisture may change, and it may change over season, it may change over weeks, it may change over days, and that's why it of course affects the phenology of the plants. That is when they are flowering, when they are fruiting, when they are shedding their leaves, all these are concerned. So soil moisture will also tell us about the weather forecast, about droughts, floods, crop productivity, and water, energy, and carbon cycles, and of course about the climate change, because it is related with the carbon cycles. Samples should be collected from different horizons, preferably from the 5 centimeter, 10 centimeter, and 15 centimeter depth in a pre-marked and pre-weighed containers, and should be brought in the laboratory and should be weighed properly, and they should be kept in a oven for drying at least for 24 hours. The dried soil with container should be again weighed, and the calculation, one can calculate the percentage of the moisture. We have to calibrate the scale or the balance according to the manufacturer's direction. If we are using an electronic balance, check the balance is measuring in grams and it is zeroed properly, and record the mass of each container without the lid or within the plastic bag to the nearest 0.1 gram. Label each container properly for the mass of the container or the plastic bag, and of course we have to mark the container properly so that we can know that which container contains which kind of the soil. So one can calculate the percentage of the soil water content that is wet sample minus dried sample divided by dry sample minus empty canes will give you the soil water content, and multiply it by 100. So that will give us the percent water content in the soil. Now, another very important parameter is soil pH. Why we want to know about the pH? Because the pH of the soil will tell us that whether the physical, chemical, and biological properties of the soil are good enough for the plant growth or not. Soil pH affects the amount of nutrients and chemicals that are soluble in water, and therefore the amount of nutrients available to the plants. Some nutrients are more available under acidic conditions, while others may be available under alkaline conditions. In general, roughly 14 to 17 essential plant nutrients are obtained by the plants from the soil. And most of the minerals and nutrients are more soluble or available in the soil when the soil is either slightly acidic or neutral or a slightly alkaline form. So we can say that most of the agricultural, horticultural crops will grow very good if the pH is 6 to 7.5. The 7 being neutral, as we go down from 7, it is increasingly towards the acidic side. And if we go up from the 7, it is increasingly towards the alkaline or the basic side. So what we need is, for pH, dried and sieved soil, pH data sheet, distilled water, pencil or pen, 100 ml graduated cylinder, glass string rod or string device, 400 ml containers, pH meter or pH paper, or we use pH pen in globe, and of course a balance to up to the accurate to weigh the soil up to 0.1 gram. So for measuring the soil pH, the soil should be 2 point, very important, it should be dry. Second point is it should be sieved. It should not have very big pebbles or the big particles in this one. So what we do is we mix equal amount of the sieved soil with equal amount of water, double distilled water preferably, that is 1:1 ratio. And then we stir the soil and water with the help of a spoon or any kind of a stirrer or a glass rod for 30 seconds. Keep it for some time. Again, we mix it. And then after 3-4 such kind of mixing, we allow the water solution to settle down so that all the soil particles will settle down. And then from the upper liquid or supernatant liquid, with the help of a pH pen, we can take the reading. Now pH pen should be very well calibrated before you start the taking of the reading, and then we can take the samples from different origins, and we can repeat the same experiment 3, 4, or 5 times to know the exact pH. This is the stirring of the soil for some time, and this is the pH meter. Now the soil carbonates are very important. Most common type of the carbonates are either dolomite or less type of the carbonates. They may include sodium bicarbonates or the ferric or the ferrous carbonates. Now the carbonates buffer the soil pH and are indication of the relative abundance of the bases or the alkaline bodies. Carbonate mineral plays important role in soil management. Distribution and amount of the carbonates influence soil fertility, soil irradiability, and the available water capacity. And that's why knowing the carbonate is very important. In general, the carbonate, when treated with any dilute acid, they give effervescence. In globe, instead of the any kind of the acid, we use vinegar or acetic acid for the purpose that students are involved, and this acid may not affect the students. And we can then measure the presence or absence of the carbonates, as well as we can know the quantity of the carbonates, that is whether they are present in very poor amount or medium amount or very high amount. So this is how we can measure. So this is how the soil carbonates and the soil pH is being done. Now then we come to the very important topic, that is IVSS, that is International Virtual Science Symposium. How students and teachers together can help in making certain small research projects, and how they can use the scientific knowledge in some research projects. Knowledge about the environment should be shared in easy ways without barriers. The IVSS is an online scientific space where students from all scholar years, including primary grades to the universities, can share their scientific findings in 4 investigation areas of the globe. The participation is open to all globe countries, and the results are available to all the globe countries and students and teachers and scientists. The GLOBE program is contributing to the increasing of the awareness of our planet through support for research projects that include the collaboration between all GLOBE community members. And it includes students, teachers, and science, technology, engineering, and mathematics professionals. Science is not only about what you discover, but also about how you communicate your discoveries to transform data into valuable information that could be useful to understand in a much better way what is happening in our planet. Though IVSS has been helping us all to share ideas and research findings through discussions, opportunities, and has strengthened collaborations between students and scientists. So one can take different kind of the scientific investigation projects. Now for undertaking scientific investigation projects, first is that one should conceive the idea or the hypothesis, that what kind of hypothesis is there. Suppose we want to understand the relation between temperature and nutrient uptake, or relations between the temperature and the water, soil moisture content. Then design our experiment according to our hypothesis. Then we have to make many observations and take readings sincerely. Try to avoid observations or the experimental errors. Compute your data and try to prove the hypothesis in a correct way. Write your IVSS report under the guidance of your teacher, and in case you need, you can take help, guidance from the university, college, school teachers of your area, and then you conclude your findings independently based upon your observations and your readings and your data which are available. Now, some of the projects which one can undertake, say soil texture and water holding capacity. Students can mix different ratio of sand, silt, and clay and make 6 to 10 types of combinations. Measure the water holding capacity of these different soil. Another is same different kind of soils and plant growth parameters. Now the similar kind of plants are grown at the similar time with the similar kind of the water they give, and they can take the different parameters like number of branches, number of leaves, number of fruits, number of seeds, total dry weight underground and above ground, and in this way you can study the effect of soil texture on plant growth. Similarly, you can also understand that the different kind of the soil texture under a tree, in a forest area, in an agricultural land, which is under cultivation, in a garden land, in a big lawn, or in a big, garden, or in a grassland, and in different kind of the land use places. Similarly, soil pH can be studied in different type of the soils like garden, agriculture, school area, grasslands, forests, etc. Study can be undertaken about the soil microorganisms and the macroorganisms, relationship between soil texture and its water holding capacity, relationship between air temperature with soil temperature, and soil covered by herbaceous vegetation, trees, grasslands without any vegetation, not or the land covered with the litter and directly exposed to the sun. Here is a photograph where the students from India, they are showing some scientific model to Dr. Deshbandhu. So the soil texture and structure near a river bank, near a lake, in an agriculture field, in a forest area, in a grassland, in an urban and rural areas, places where solid waste has been disposed of can be undertaken. Effect of industrial waste on the soil pH, difference between pH and rainfall, water and soil, change in soil moisture and pH value in addition by addition of the vermicompost, effect of agriculture residue, waste in the soil, effect of blue-green algae growing in a paddy field, and its change on the soil characteristics. So what we study actually plant, soil, and microorganisms interactions. That is important for any kind of the students' projects. Food web can also be worked out. First trophic level, that is photosynthesis, etc. Second trophic level, that is decomposing the pathogens and the parasites. Third trophic level, fourth trophic level, fifth trophic level, different kind of the nematodes, fungi, arthropods, birds, they can be studied. We cannot, if some students cannot study the whole food web, one can understand only a small food chain, or maybe that what kind of the different microorganisms are present in different soils growing different kind of the crops in the same area. Similarly, nitrogen fixation is being done by the cyanobacteria, and students can study the nitrate nitrogen in an agricultural field or a paddy field before and after the growth of the blue-green algae or the cyanobacteria. We know that roughly 20 to 30 kilograms of nitrogen per hectare is fixed in by the blue-green algae in agricultural field. Azolla is another field fern which also fixes atmospheric nitrogen and is a good source of atmospheric organic nitrogen and enriches the soil. So one can study the nitrogen before and after the growth of the azolla in a particular field. So this is all about the soil and the different studies one can undertake. I thank everyone for their